Hey everyone, thank you all for coming to this webinar. Today we have the privilege of having Syma, Khalid and John Essex, both professors at the University of Southampton, here to talk to us with their HEC Biosim hats on about the work that the HEC Biosim Consortium has been doing on researching, especially molecular dynamics researching of the SARS-CoV-2 virus. I believe, from my understanding, this is going to be a two-part talk with Syma giving her presentation first, followed by John. And with that, Syma, if you'd like to take it away, please go right ahead.
Sure, thank you, Julian. Well, thank you, Julian, and the team at ARCHER2 for the invitation to speak to you today about some of the work we've been doing, as part of a wider UK simulation community, and then later on John will talk specifically about work that's been done within his own group, where the work we're talking about is all targeted towards fighting the horrendous COVID-19 infection that seems to be taking over all our lives at the moment, and in particular where supercomputers have been used within our studies. So I thought I'd start off simply by introducing HEC Biosim first and foremost. So HEC Biosim is an EPSRC funded consortium. Primary focus is biomolecular simulations. So these can be Monte Carlo simulations, molecular dynamics, but they are simulations at the molecular scale where our focus is biology. So they're biologically driven. And as a consortium, we provide training for high performance computing best practice, but perhaps most importantly, we hand out time on Archer, and that's soon to be ARCHER2, but also a bunch of tier 2 resources, which up to date has mostly been the Jade 2 GPU facility based over at Oxford. So the consortium is led by a management committee, which consists of academics from around the country who have an interest in biomolecular simulations, but use a diverse range of codes and lots of different methods. And I think we fairly well represent the broad spectrum of techniques and codes that come under the umbrella of biomolecular simulation. So essentially people apply to us for time on facilities. I then put together a panel of 3 or 4 people from the committee, plus an external member of the committee, and James Gebby-Rayet, our technical support, and we then evaluate all the proposals and pick the ones that we think should be supported. And when the pandemic first hit, we were approached by Archer to set up a panel whereby we were provided with proposals for using high performance computing time, in particular Archer, and then later on Jade, to go through those proposals and take a look and decide which ones were perhaps scientifically most sound. And what I'd like to do today is go through with you some of the results that have been coming in through work done on those resources. But actually, because we're, I think, quite a general audience, I think it makes sense, before diving into the details of the science itself, maybe it's good to just have a bit of an overview of the SARS-CoV-2 virus. So first of all, we'll start right at the beginning. The virus is called SARS-CoV-2, and this causes the infection, which is called COVID-19. As I'm sure many of you know, viruses cannot replicate by themselves, so in essence they're not really alive as such. They need a host in order to achieve replication. And if we take a molecular view at a virus particle, because essentially that's what they are, it's a particle, and we take a slice through the middle, it would look something like the diagram you can see on the left-hand side. And essentially what this is telling us is that the virus particle is roughly spherical. It's surrounded by a layer of lipids, and so it's called an enveloped virus. It's enveloped by this layer of lipids. And for those of you who are not familiar with the chemistry of lipids, essentially it's a greasy layer. It's not water-like, it's more oil-like. There are some proteins that are embedded within this layer. So you can see the spike protein sticking out from the virus in orange in the diagram there. And this protein sticks out all around the virus and creates a sort of crown, thereby giving this class of viruses their name, the coronaviruses. These proteins are the workhorse of the virus. So in a way, you can think of them as the workers or the builders. They do the sort of building of new virus particles. Now, inside that greasy layer, and you can see in red in this particular diagram here, we have RNA. So this is a nucleic acid which contains the blueprint to creating new viruses. So it contains all the instructions needed to create a new particle. The problem is the virus can't do that by itself. It needs us to do that. It's got the instructions, but it can't actually go ahead and make the proteins by itself to go away and replicate. So what it does, and it's evolved to do this, and actually if you think about it too much, it's pretty frightening actually. The problem it has is both the viral particle and the host, in other words, our cells, but when I say host, I mean us, both our cells and the viral particle are surrounded by this greasy lipid layer. Now, the problem the virus has is it has to get inside our cells in order to replicate itself. It can't do that by itself, it needs us. So how it does that, and if you take a look at this diagram here, firstly, I'll just specify that the virus bits are in orange and our bits are in gray. Okay, so the virus particle, what it has to do first and foremost is the spike protein, so the one that gives it its corona name, this thing that's sticking out all over the place that I showed on the previous slide. The viral spike protein has to latch onto a protein that's embedded in our membranes. And this happens to be a protein called the ACE2 receptor. Normally, the normal function of the ACE2 receptor is that it's used, our bodies use it for blood pressure regulation, right? So it has its own function. But the virus sees this protein, it recognizes it, and latches onto it. Okay, so now what's happened is the spike protein has latched onto the ACE protein, which is in our cells. Then another one of our proteins, which is an enzyme, comes along and it chops the head of the spike protein off. Right, a chemical reaction occurs, bonds are broken, and the head of the spike protein comes off. And inside this spike protein is essentially compressed in like a spring is the fusion machinery. As soon as the head of the spike protein comes off, this fusion machinery is released. And you can see this in the orange squiggly lines on this next diagram. So what happens is this fusion machinery, which is also part of the spike protein, it's still protein, it comes out like a spring, it springs out of the spike and embeds itself into our membranes, so that greasy layer that's in our cells. What it can do now is it can bring the two membranes together, so the viral particle, the greasy layer on that particle, and our membranes come together and fusion occurs, and that opens up a channel. Through that channel, the viral particle, so the viral RNA, sorry, enters our cells. Okay, so what's happened is the spike has latched on, it's caught hold of one of our receptor proteins, another protein has come along, it's snipped the head of the spike off, stuff has come out of the spike, which has enabled both the viral particle to fuse with our cells, and then the RNA, so the blueprint from the virus, has gotten into our cells. And all of this takes about 10 minutes of real time. Okay, so now the virus is inside us. What happens next? Once it's inside our cells, that RNA hijacks our cellular machinery to produce proteins. So our body, our cells read the RNA thinking it's part of our own cells and start converting that, translating that RNA into proteins. These proteins, as I mentioned earlier, are the workhorses. They're like the builders, right? They perform a range of functions, including producing more RNA, and so they represent a target for antivirals. Okay, so without these proteins, the virus can go no further. Now that these proteins have been produced, they go about doing different tasks. They assemble new viral particles using a whole bunch of multi-step process. They take the RNA, they package it all up, they create a lipid layer around themselves, and so package it all in to create new particles. Unfortunately, it turns out that it's highly likely some of our own proteins also help to make the new viral particles. They get hijacked. So once the new viral particles are formed, in other words, once new RNA has been formed, the proteins have packaged it all up, they've surrounded it all by a lipid layer, then they burst out of the lung cell, leaving the lung cell for dead. Okay, so they've replicated themselves using our own machinery, and have left the lung cell now, and that lung cell is dead. And that process takes about 10 hours. So you can hopefully see there that without us, the virus is essentially, it can't come to any harm. It can't really do anything. It can't cause any damage. It needs us for that. Okay, so if we're going to fight the virus, if we're going to fight the infection it causes, then how are we going to do this? There's two ways. We can develop vaccines, so we can prevent the infection in the first place, or we can develop, and in addition to that, we can develop antivirals, where antivirals will treat early infection. So you've already been infected, but the antivirals can help then combat the infection. To create a vaccine, what we need to do is we need to stimulate the development of antibodies. We need our immune system to become stimulated to develop antibodies, and essentially what those antibodies will do is where they will prevent the spike from binding to the receptor in our cells, or prevent the spike from releasing the fusion machinery. In other words, they've got to disable the spike from carrying out its normal function. Whereas antivirals will also either interfere with the spike, or they'll interfere with other proteins that play key roles in the viral life cycle. Those proteins can be ones that exist in the membrane of the virus, or they can be proteins that are only produced once the virus is inside, has gained entry into our cells. So the questions that our community, the biomolecular simulation community, are interested in, and that is questions that are essential for us, what's the structure of proteins that we're interested in? Important proteins, what do they look like? What's needed to prevent that protein from functioning correctly? And to answer those questions, so a follow-on question from that then is, where should drugs bind? Where on the protein? We might know what the protein looks like, but we also need to know, where should drugs bind? Where are the likely sites? Of course, beyond that, there are lots of other questions too. I don't want to, pretend this is a very simple scenario. There's lots of questions surrounding drug toxicity, etc. But I guess those are usually questions that the medicinal chemists will address, rather than at this level molecular simulation folk. So what I'm going to do in the next few slides now is go through with you areas where the UK biomolecular simulation community, funded by HEC Biosim, have been working to address some of the questions in this orange box here. So the first question we might ask is, if we think about the spike protein, okay, we know we've got to somehow stop that spike from binding to our receptors, or we've got to stop the spike from releasing its fusion machinery. Okay, we know that, but where are the target sites on the protein? Where can drugs bind? So there's been a lot of work in this area from the UK community, and I'm going to highlight 3 cases here, but that doesn't mean that these are the only 3 the only 3 efforts in this area. There are a bunch of others. These are the ones where funding has come directly from HEC Biosim. So some work in Michele Vendruscolo's lab at the University of Cambridge. They've been using state-of-the-art enhanced sampling methods to look at how the spike protein opens. Now this is kind of, I guess you could say, well, we've got crystal structures and microscopy images of spike proteins, so why do we need to do these simulations? Why do we need to do these calculations on a supercomputer? Well, the answer to that is that these images don't show, the spike protein is covered in sugars. These images don't show the sugars. Also, they capture the protein in one state or the other. They don't show the transition between them. They don't show us where there are any hidden binding sites. And so calculations done that, for example, like the one that's produced this movie here, have been done on ARCHER, where calculations have been run for weeks to produce this image, where we begin to get an idea of how the protein opens and therefore where target sites might be. In a similar vein, some work done in Southampton and at the A-STAR Research Institute in Singapore. So collaborators here was a project really led by Peter Bond in Singapore, collaboration with my own group and also Professor Max Crispin at the University of Southampton, where we've been looking at the sugars, which you can see in yellow here, on the spike protein, and trying to work out where are the binding sites, how do the sugars affect where the drugs might go. These are huge simulation systems. There's 1.8 million atoms, and for those of you with an interest in performance on ARCHER, then these simulations will, using 20 nodes, so therefore 480 cores, will give us about 13 nanoseconds per day. So these simulations have been run over months, and we really needed ARCHER support for this. There was nothing else in the UK that could provide us with this level of compute. Work done in Bristol in the group of Adrian Mulholland has also looked at the spike protein, where here a cryo-electron microscopy detected linoleic acid, a greasy molecule essentially bound to the spike protein, which was unexpected. Simulations from Adrian's group, again using ARCHER. These are 680,000 atom simulations. You can see here in the spheres, you can see the linoleic acid. So this molecule, it's a greasy molecule, it's stuck inside the spike, and it turns out that it seems it may even be stabilizing the spike protein, and therefore represents a good target site for drugs. So what you would do is you take a look at that site and say, actually, if I can design a drug that binds into that site, that's probably a good place for it to go. And this work has been published in Science earlier this year and has achieved also recognition in the mainstream media. You can see here the Evening Standard and the Daily Mail. So once we know where to target the spike, then what do we do? Just like to highlight two projects here for you. Again, we've got Michele Vendruscolo's lab at the University of Cambridge. So Michele's group are world experts in designing antibodies and nanobodies. So they have done these are, as you can see, smaller simulations, a bunch of smaller simulations using the Dyad GPU cluster, where they've taken two types of nanobody. One's derived from llamas, and that's a collaboration with Jim Naismith's group at the Rosalind Franklin Institute, and also work from Dave Stewart. And secondly, completely computationally designed nanobodies, and they're looking at doing a bunch of simulations. You can see here to look at how the nanobody's in blue and the spike protein is in grey, and you can see how does the nanobody bind to the spike protein. So typically one would look at the interface, the region of binding where the protein is making contact with the nanobody, and try and improve that binding. So you look at the amino acids that are involved and look at their interaction and use chemical knowledge to try and improve that binding. And this is where the simulations are absolutely essential. Again, targeting the spike is a collaboration between Francesco Gervasio and Josep Haider, both working at University College London. So they've taken a slightly different approach. What they're doing is saying, well, we know the spike, we need to prevent it from binding to the ACE receptor protein, so why don't we physically occlude that region? And so they've designed their own peptides. So you can see here two helices in the sort of dirty greenish color at the top. So they've got constructs of two helices and 3 helices, which they've designed using a machine learning approach and free energy calculations. So they take these peptides and they say, well, actually, if these are therapeutic peptides, if these are administered, then what they'll do is they'll sit at the interface between the receptor protein and the spike. And so the spike will not be able to recognize and bind to the receptor, and therefore the other enzyme cannot come along and chop its head off and all the fusion machinery come along. So they're using a sort of, if you like, almost a brute force physical approach. Let's prevent this interaction from occurring. It's a nice study because they're using both machine learning methods and free energy calculations. And these are simulations that they've been doing both on JADE for the smaller systems and on ARCHER for the larger systems. There's a bunch of other proteins in addition to the spike that are targets for antivirals. As I've mentioned already, these proteins are the workhorse of the virus. And another study, which is being done again by Francesco and Shozeb at University College London, is they're taking a look at the NSP1 protein. And this protein is it's interesting because what it does is it suppresses our innate immune function. So it prevents us from responding to the virus. So clearly we want to stop this protein functioning. We want to knock it out somehow so our immune response can deal with the virus. And so there's a structure of this protein, but we don't know where we would target it. We don't know where you would target drugs to the protein. So they've taken a nice approach where they use water and benzene. They flood the protein with water and benzene in an attempt that the water and the benzene will enter different binding sites and open up hidden binding sites. And again, they're using JADE and ARCHER. Finally, HEC Biosim has been able to help set up a cross-Europe consortium where a bunch of scientists are looking at a protein called NSP13. This enzyme translocates RNA, so it's really important for processing and packaging up the viral RNA. But to understand this is difficult because you have to simulate the protein, you have to simulate the protein in combination with the RNA. There are no real conclusive X-ray structures, so a lot of modeling's involved and important. And also it turns out there are ions needed in key regions. And for those of you who are not experts in molecular dynamics, I can assure you that the expertise needed to simulate proteins are quite different from the expertise needed to simulate and model RNA. And then it's quite another thing on top of that to work out ion binding sites, for example. And so expertise were needed from a bunch of different scientists. And it was really nice to be able to put together as a consortium this bunch of scientists from King's College London, so Edna Roster, University of Leeds, Sarah Harris, Jeff Wells at University College London, and a whole bunch of other scientists here. So Andre at Dundee, Nadia at Berlin, and Elisa at Paris. So they've all come together to sort of put together their expertise such that I can just happily show you this little movie here, where we now have a working model of the enzyme bound to RNA with ions in. And I'm happy to report that they are now working with experimental colleagues who are taking forward some of their ideas of what drugs should look like that bind to this protein. And this consortium used ARCHER time. There was some JADE time used too, I think. And what was really nice was that the University of Paris also facilitated by providing time on the French facility. So it was really nice that this effort that was kick-started by EPSRC then brought in resources from elsewhere to sort of, as a community, take this project forward. So finally, I just wanted to finish off by saying there was, I focused on JADE and ARCHER, but there has been also facility provided through DIRAC, based over at Cambridge. And Julia Michel at the University of Edinburgh has been doing some work, again, looking at the spike protein with stapled peptides. Martin Umschneider at King's College London has been trying to design antivirals that will target the viral membrane. I don't have time to go through these in detail here, but I wanted to acknowledge that DIRAC have also provided high-performance computing facilities. And I guess I wanted to finish by saying that these projects that I've highlighted to you fairly quickly, I've mentioned only the principal investigators, but there have been a whole host of postdocs, PhD students, research assistants, technical staff involved. And in many cases, people have moved off the main project they were working on to focus for a while anyway on this efforts towards COVID-19. And when you're doing a PhD, which is 3 or 4 years, or a postdoc, which can be between 2 to 4 years, then changing projects is something actually that's not very easily done. So I just wanted to pay tribute to people who've done that and who've worked in a community-minded way, but actually also tension that very well with also looking after the interests and the career interests of the individual people involved, because after all, there is going to be a life after COVID, and people will go on to careers, and so publications and outputs from these efforts are enormously important. And I think the community's done a fantastic job in tensioning what's needed for the community with what's needed for the individual, and that's been fantastic. And finally, I'm going to finish off with some acknowledgements to ARCHER, not only for providing us with time, but providing us with the technical support and help throughout. I'd also like to acknowledge Jade for likewise providing time and technical support, but actually also ATOS. So ATOS are the suppliers of Jade. They also provided us with compute resource outside of the Jade facility. So I wanted to acknowledge that too. And last but not least, EPSRC for the direct interaction right at the start of the first lockdown, and providing us with a lot of this resource and actually giving us a lot of freedom and trusting us to know that we knew the science best and just to get on with the science. And that's been absolutely fantastic. And with that, I'm going to stop talking and hand over to my colleague, John Essex.
Okay, so thank you, Syma. That was an excellent overview of the work that's being done in the community, largely facilitated by the UK's investment in HPC through resources such as ARCHER and Jade. So we're, as a community, we're really grateful for that investment. What I'd like to do today is describe some work that's been done jointly in my lab with Chris Lawrence's group at King's College London, where we've been doing some simulations to explore the binding sites of one of the SARS-CoV-2 proteins, specifically the main protease. So what I'd like to just kick off with is just emphasizing the fact that this work has been substantially done by others. That's usually the case when I give a presentation. Obviously, as the group head, I do precious little direct running of calculations these days, but it's particularly true in this case. So the study I'm going to show you was designed, conceived, ran and analysed and written up by 2 PhD students, one in each of our groups. So Khalid works in Southampton, and Mohammed Ali works at King's College London. So they ran with this all by themselves. Chris and I only became involved at the end when we were discussing the final preparation associated with the manuscript. So this is a remarkable achievement for pair of graduate students, and but they really did do everything associated with this, and to be honest, they probably should be presenting this now. I noticed that Mohammed Ali is in the session, so welcome. And if I get stuck with any questions at the end, I may very well turn to you to help me out. But anyway, just to reiterate, Mohammed Ali and Khalid did the overwhelming amount of work that I'm presenting here, as I say, including conceiving of the project. So what are we looking at? So as Simon's already indicated, there are 2 main protein groups that we might wish to target in terms of trying to treat SARS-CoV-2 and the COVID that arises from that, the Covid-19 disease that arises from that. One of the structural proteins, of which the spike glycoprotein is one, and Simon's already illustrated to you some of the approaches that are being used in terms of small molecules and nanobody design to target those, and of course the vaccines that are now showing such promise also are designed to bind to the spike glycoprotein. The second group of proteins are associated with the virus life cycle, and they're expressed on infection, and one of those is the main protease. So as I've already indicated, the former group, the structural proteins, are targeted for vaccine development by disrupting their interactions with the ACE2 receptor and thereby suppressing host cell infection. But we're targeting specifically the main protease, and we're asking the question, could small molecules bind and inhibit the main protease? And the main protease is involved in taking the polypeptides, polyproteins, sorry, that are synthesized as a result of transcribing the RNA that the virus injects in the cell, and cleaving those up at a number of known sites to create the proteins that then go off and do the work that's required to ensure that the virus replicates and spreads. So if we were able to inhibit the main protease, we would hope to be able to stop the virus replication process in its tracks. Now, protease inhibitors are well known. They are widely used, for example, in the treatments of HIV/AIDS and hepatitis C. So targeting proteases is something that the pharmaceutical industry does well. In our case, we're also seeking to see if we can assist in that design process using computer simulations, and particularly molecular dynamics simulations in this case. And these have been effective for inhibitor design. For example, they were involved in designing HIV integrase inhibitors, and also the N3 covalent inhibitor of SARS-CoV-2 that's recently been proposed a few months ago. So our objective then is to see whether simulations can assist in the design of inhibitors for the main protease. So if we just look at the main protease structure, this is a figure I've taken from a bioarchive paper by Annie McCammon, Romeo Morrow, and Tara Stein. And they've been using enhanced sampling technique called Gaussian accelerated molecular dynamics to try and explore for potential binding sites in the main protease dimer. And they essentially identified three possible regions where you could develop potential inhibitors. Obviously, there's the active site, which is where the polyprotein created by the RNA within your cells is cleaved, so that's the active site. There's also the interface between the two monomer units in the dimer that one could conceivably target. And there's also a distal site at the back of the molecule with potential role as an allosteric effector that one might choose to target. But our primary interest was in trying to look at binding to the active site. Now, in relation to that, we're not working in a complete vacuum, as it were, because SARS was first reported, obviously, in the early 2000s. That was a respiratory infection that arose in China, again, spread further in the world. It was somewhat less infectious, thankfully, but somewhat more lethal, which is obviously not a good outcome. So there has been an effort to design inhibitors for SARS-CoV-1. And the structure on the left-hand side, you can see in the figure. If I just try and get my laser pointer working. Nope, I've gone too far ahead. Let me just see if I can get a laser pointer. Can everybody see that?
Yeah, we can see that.
Yep, brilliant, thanks. Okay, so the image on the left shows one of these ligands to SARS-CoV-1, which is called DTF, and that's a moderate binding ligand, micromolar if my memory serves me correct. And it binds to the active site in the way shown here. So the protease works by cleaving the peptide backbone of the polyprotein, and it does so by this cysteine engaging in a nucleophilic attack on the amide group. And that cysteine is primed by hydrogen extraction through to this histidine. So what's happened here is that the inhibitor on binding has split the arrangement, the optimal arrangement for the interaction between the cysteine and the histidine. In doing so, it's picked up some extra interactions. So the nitro is binding to one of the groups on the histidine. So there's some sort of pi-pi type interaction between group here on the inhibitor and the histidine line below it. So this is the structure of D3F here on the left-hand side, and it's taken from this crystal structure. So we'll refer to that from here on as a hollow type interaction, because it's derived from a bound ligand structure, but the catalytic dyad has been disrupted. On the right-hand side, we have a similar figure. This is derived from simulations of the SARS-CoV-2 main protease, but from an APO-like structure. So this is a structure without a ligand bound. In this particular case, this simulation contains a putative ligand, which we refer to as Lig. So this was suggested as a potential binding partner for SARS-CoV-2 by other workers who are using a combination of machine learning and docking type studies. So they were interested in knowing, is there a potentially good binding geometry that could be observed for this ligand? So in order to determine that, Khalid and Muhammad Ali performed co-solvent simulations. So they put the ligands in solution around the protein, some in the vicinity of the binding site, and just asked the question, well, how might it bind? What might the optimum binding geometry be? And what you can see here is that on forming the complex, the catalytic dyad is well formed. There's a hydrogen bond between this hydrogen and the cysteine sulfur. So the dyad is not disrupted. So we have some indication from SARS-CoV-1 that dyad disruption might be possible, but this simulation for SARS-CoV-2 doesn't suggest that you've seen that. So we then went away and had a look in the crystallographic literature and asked ourselves the question, well, what does the X-ray record say? And obviously it's still very early days, but of the 192 structures that Khalid was able to find in the PDB that were of the SARS-CoV-2 main protease, we analysed them for whether or not the dyad was intact or disrupted. So the intact form we refer to as APO, just to reflect the fact that it's certainly found in cases where there is no ligand bound. And hollow just reflects the fact that the dyad broken structure was identified from a SARS-CoV-1 structure with a particular inhibitor bound. So really, we should perhaps more clearly label these as intact dyad and disrupted dyad. If we look at the histogram and the distances, then you can see that the majority of structures that are in the crystallographic database for the CoV-2 main protease have interaction distances between the nitrogen and the sulfur here of around about 3.5 to 4 angstroms. So that's indicative of a weak hydrogen bond. So just histogramming the distance tells you that, and you can see that there is another bump out here at a sort of 5-6 angstroms, which is associated with a weaker interaction between the cysteine and the histidine. So the majority of the structures are associated with the intact dyad, 97%, very few associated with the disrupted dyad, the hollow type, only 3%. So this would suggest then, if one were to use the crystallographic record to guide inhibitor design, if one were to use virtual screening, for example, then one might be tempted to just use the intact dyad structure as your example, your seed structure, as it were, in order to incorporate the ligand, potential ligands into the binding site. And you might therefore miss the opportunity to exploit some interactions if you fail to consider the possibility of this disrupt dyad type configuration that we're seeing on the left-hand side. So what we wanted to try and do through simulation is try and see if we could determine the relative free energy differences between this sort of dyad intact and dyad disrupted conformation, and see whether or not the free energy cost associated with dyad disruption could be recovered through ligand interactions. So in order to do that, we turn to enhanced sampling calculations. So this slide is taken from specifically trying to address examples of studying protein folding, but the same principles apply here. So we're looking here at trying to sample on an energy hypersurface, as indicated by the figure on the left-hand side, where we have minima separated by barriers. Those barriers may be high in energy with respect to thermal energy, and therefore barrier crossing events are rare and your sampling is restricted. So what one can do then is one can project these, this complicated energy surface onto a few collective variables, which could be linear combinations of Dihedral or Cartesian collective variables in the system. And then we sample and project our simulation onto that. So this figure here is just showing a normal molecular dynamics calculation taking place on a particular collective variable. And in normal molecular dynamics, it's not uncommon for your system to be trapped. because you have insufficient thermal energy to cross the barriers. So essentially you sample very little and you just sample the local minimum. But what we'd like to do then is we'd like to apply biases to force your system to leave its local minimum and sample the entire energy surface. And this can be achieved, for example, using temperature. But there are other techniques that one can use as well, rather than just simply adding kinetic energy to get the system to sample outside its local minimum. The particular technique that we've used here is a technique called metadynamics, which was first proposed in the early noughties. But essentially, the way metadynamics works, for those of you who aren't simulators, is the system identifies regions of the collective variable that it's sampled, and as it samples those regions of the collective variable, it lays down biasing potentials, which are Gaussian in character, which are intended to push the system away from regions where it's been. So the notion is that you drop piles of sand where you've simulated, and in doing so, you push yourself out of your local minimum to sample more of the collective variable. So as the simulation runs, then we accumulate bias, we build up these piles of sand, we fill the various minima, which enables us to swap to adjacent minima, and so on. So as the simulation runs, we get accumulation of these biasing energy terms until ultimately one samples uniformly and consistently across the entire collective variable. Once one has done this, one's filled in all the holes, as it were, with the sand, it's possible to take a negative image of the bias that you have obtained to get the free energy surface. So in blue here, we have the biasing potentials that are associated with this particular energy surface, and the negative of those biasing potentials is the underlying free energy surface that we're able to recover. So by applying this process of metadynamics, one would hope to be able to get some feeling for the relative free energies of the open and closed forms of the protein, of the binding site, sorry. So in order to do that, we would like to try and, give me, I've stepped ahead, we would like to try and sample the backbone dihedral angles, sorry, associated with the histidine that's binding to the cysteine. So by looking at the structures of the main protease 1 and main protease 2 from SARS-CoV-1 and SARS-CoV-2, we could see that the main discriminant in terms of the intact dyad and the disrupted dyad type structures were associated with this side 2 backbone triangle, which I've indicated on the right hand side here of the figure. So this dihedral angle will be the ideal one that we would want to apply enhanced sampling to in order to determine the relative free energies of the conformations. Now, unfortunately, when they tried to run this, Khalid and Muhammad Ali discovered that this was not a good choice of collective variable. The simulations did not perform well. They showed some instabilities. So as a result, they then tried the alternative, which is to look at sampling the Psi1 dihedral angle illustrated here on the left-hand side. Their intention there was to see whether or not by sampling this they could disrupt the dyad and indirectly bring about enhanced sampling of Psi2. Essentially, if you break the hydrogen bond, then Psi2 will be free to move, and we can get our statistics just looking from the analysis of the rotation on the Psi2 dihedral angle. So the data on the right here shows the free energy curves that they obtained from their metadynamics calculations for sampling the Psi1 dihedral angle, this one here on the left-hand side. And they essentially identified 3 minima from these calculations, which are shown by the purple, cyan, and yellow regions on these this free energy surface. And over the course of the simulation, they monitored the difference in the energy of the minima with respect to each other in those 3 regions. And that's illustrated in the 3 figures below that. And those 3 figures are showing that the difference in the free energy of the minima are sort of stabilizing at around about small values, well below 2 kilojoules per mole. But those curves and traces are largely stable at around about 70 nanoseconds. So these calculations seem to be converging reasonably well, which is good, useful, and important, of course. So having got enhanced sampling, we can just illustrate the results that we see by just doing a little movie. So the figure at the top left-hand side here shows the intact dyad. This is an example of a broken dyad structure, sorry. And on the right-hand side, we have a, oops, let me just go back. We have a movie showing the metadynamics calculation, so it will zoom in on the active site in the main protease, And you can see that that histidine is now being sampled extensively. This is over the course of the metadynamics. The biases are being applied specifically to the Cy1 dihedral, but we're also getting movement in the Cy2. So it's having the desired result in enhancing the sampling in this particular system. In terms of how we proceeded then with the analysis, or rather how they proceeded then with the analysis, to reiterate, we're biasing within Cy1 here on the left-hand side, and we're reporting the distributions that we're seeing in Cy2 that arise from that. So in this plot here, we're looking at the angle that we see in that dihedral angled Cy2, this one here. So in green, we have the distribution that we obtain in a normal molecular dynamics simulation with the Lig co-solvent present, and what that is illustrating to you is that the dyad is stable, it's not being disrupted, the Cy2 dihedral angle is not changing. Over here in blue, we have the equivalent distribution for the SARS-CoV-1 case with the D3F ligand bound. Here the dyad is disrupted, and that too is stable. We're not seeing any sampling. And then in purple, we have the metadynamics calculations on the SARS-CoV-2, but with no ligand bound, and we can see that we're sampling both conformations. We've got a purple peak here, which is associated with dyad formed, and a purple peak here, which is associated with dyad disrupted. We're then able to look separately at the hydrogen bond distance between the sulfur atom on the cysteine and the nitrogen epsilon on the histidine, and we see similar results. So in the case of the dyad disrupted simulations, which are here in blue, we see a long hydrogen bond distance. In the dyad intact simulations, which are on the CoV2 without a link with the co-solvent link just outside, we see that that is intact and stable at a hydrogen bond-like distance. And then looking at the metadynamics calculations, which is the purple underneath, we can see that we're switching between the two different types of interaction, intact and disrupted. We can analyze these data further by projecting onto a two-dimensional surface. So on the left-hand side, we have on the x-axis the Cy2 dihedral angle, and on the y the hydrogen bond distance. And we can, this structure here, these simulations here are from the CoV1 with the D3F inhibitor bound, and we end up with a disrupted dyad, a dihedral angle of about -0.5 radians and a hydrogen bond distance of 7-8 angstroms. On the right-hand side, we have the equivalent calculation on the SARS-CoV-2 APO. So here we have an intact dyad. We have a dihedral angle of about 0.5 and a distance of somewhere between 3 and 4 angstroms. And then in the middle, we have a single repeat of the metadynamics calculations, where we see both the dyad intact and dyad disrupted form. You'll notice the dyad disrupted form is somewhat shifted with respect to the figure on the left-hand side. That's because we don't have a ligand in. We're just applying the enhanced sampling. So we see a reduction in the hydrogen bond distance, and that's because we get some reorientation of this cysteine. The cysteine is sort of trying to fill the hole and trying desperately to form some interactions with the histidine, despite the fact that histidine is moving out of the way. So we can then take these trajectories, and then from those we can obtain the underlying free energy surface across the Cy2 dihedral angle. So here we ran 4 replicas. So on the figure on the right here shows the aggregated data for the 4 replicas, which have been clustered. We identified 3 clusters from here, which correspond to intact dyad, disrupted dyad, and some intermediate structures which have comparatively low populations. We're able to post-process these data then to get the relative free energies between the Cy2 dihedral angle at +0.5 radians, which is associated with an intact dyad, and -0.5 radians, which is associated with the disrupted dyad. We find, so the data corresponding to that is the purple line. The variance on that, the standard deviation, sorry, on that data is in purple, the purple shading. So that shows the level of variability that we have over the 4 replicas. And you can see that the difference in free energy between the intact dyad and the disrupted dyad is of the order of 5 kilojoules per mole, so of the order of 1 kilocalorie. The blue hashed line is the equivalent distribution that we obtained from the SARS-CoV-1 simulation with the D3F ligand bound. You can see that it's just staying over in this region, and the D3F binding is stabilizing that particular conformation. So what these data suggest then is that there is the apo to hollow transition that we observe in side 2, the difference in free energy between the two conformers is of the order of 1 kilocalorie per mole, which is eminently recoverable by ligand binding. So in other words, with an appropriate ligand that binds and captures interactions with the cysteine and the histidine, it's perfectly reasonable to expect that that 1 kilocalorie per mole cost could be recovered. So just to summarize that, our simulation suggests that to disrupt the catalytic dyad in the SARS-CoV-2 main protease will cost you about 1 kilocalorie per mole. So just to re-emphasize that the potentiality for this disruption to take place was based on an analysis of the highly homologous SARS-CoV-1 protease and the data that we have associated with that, on which a great deal of work has been conducted over the last 15 plus years. So these calculations suggest that the disrupted dyad has a higher free energy than the intact dyad, but that 1 kilocalorie per mole could potentially be recovered by a ligand that binds with the appropriate interaction types to disrupt the dyad but pick up interactions as a result of that. So our recommendation then from this study is that if one were conducting a virtual screening campaign for potential ligands for the SARS-CoV-2 main protease, which is taking place at the moment, we think there's sufficient evidence to suggest that you should be looking at not only at dyad intact structures in your campaign, which is largely what's been taking place, but also there's some mileage in looking at dyad disrupted structures as potential blueprints, as it were, for ligand design in that active site. And one could imagine, for example, taking the known dyad disruptive structures from SARS-CoV-1 as acting as potential templates for your virtual screening campaign. So just to give some acknowledgements, obviously the real acknowledgement should of course be to Muhammad Ali and Khalid, who did all this work. As I say, they conceived, designed and did all the work and presented Chris and I essentially with a manuscript at the end. So so thank you to them. University of Southampton and King's College are obviously the places where we work, and particularly Southampton provided some computational resources for this project through its Iridis computer system. The work was funded by the EPSRC and the BBSRC. We are of course very grateful to ARCHER for being able to run some of these calculations on the ARCHER machine, and of course Biosim, the consortium that provided the computer time that enabled us to do this. So thank you very much.
Thank you very much, John and Syma, for the very interesting talks. I think we have time for a couple of quick questions. If any of the attendees want to ask questions, please post them in the chat. You can reach the chat by clicking the little double arrow at the bottom right corner of your screen, and I've just posted a message to indicate where the chat is. While people are thinking up and writing their questions, I was wondering, would it be possible for me to ask a quick question?
Absolutely.
Brilliant. And so I'm aware that the Molecular Simulations Society, like people working in molecular dynamics simulations and other molecular simulations, have really answered the call for this pandemic and really done a lot of impressive research incredibly quickly. I might be wrong, but I think that this is one of the first times that this has had to happen. And my question is, how far back in time could such a good answer to the call have happened within the molecular simulation community? Like, would this have been possible 5 years ago, 10 years ago, 20 years ago, and would the results obtained have been as informative or anywhere near as helpful as they are currently?
John,
okay, that's tricky.
Who's the question for? Is it for me, John, or John, or either?
Well, Syma, why don't you go ahead, because you're representing the community more than I am, Syma, so you go ahead.
Very, very nicely sidestepped, John.
Well, I didn't get to this position, without.
So no, it's a good question, and I think ever since we've had the consortium models, so you know, Heck Biosim's in its second sort of second round now. Prior to that, Adrian Mulholland was the chair, and I think wherever we've had this consortium model where we've, you know, the point has been that the scientists themselves have been able to judge the science and quickly distribute time. That has been that this would have been the case, that we can do things and mobilize quickly. The other question is, how far back would you have to go for us to provide something useful? Now, molecular dynamics, and you know, one of the caveats is always that we're limited in the timescales that we can simulate. But as John said, in early noughties, where the some of these enhanced sampling methods, well, for example, metadynamics became available, and that's really, I think they've been a game changer, really, because when you can get free energies, they're, you know, there's when either relative or absolute, they're tangible values that can be compared to experiment. So I think, you know, I would like to think that, you know, for the last decade or so, we've been at that point where we could provide something useful. And on top of that, I'll also say that molecular dynamic simulations are not going to by themselves help you work out a target, design a drug, and put it there, yeah? All the time, it's an iterative process with the experimental guys the whole time. We need structures before we can get going. So it's not just about what we can do, it's about when the high resolution X-ray structures can come along, or the cryo-EM structures. So that's all. There's also been a revolution there with what those guys can do. So I think it's a mixed bag. I don't know if does that answer the question?
Yeah, it does. Thank you very much for that answer. And sorry for putting you on the spot like that.
I don't know if John wants to add.
If I could just, yeah, I was just going to add something, just to state that I think an awful lot of what we're doing is facilitated by the technology to enable us to share data and also to do video conferencing in an event like this. So, you know, 5 years ago, it would have been comparatively difficult, I think, to engage in collaborative activities using technology like this, because it was, you know, things have come on hugely in terms of Teams and Blackboard and other pieces of software that we're all now routinely using. You know, my university, for example, deployed Teams properly throughout the institution just early this year. It was completely fortuitous. Without that, our whole teaching operation would have been significantly harder. So these sorts of technologies that enable us to do video conferencing and data sharing and so on are comparatively recent, at least in the slick and ways they now operate. Without that, things would have been a lot harder.
Yeah, really good point. Yeah, so it's not just what we can do in terms of computing and whatever. It's the dissemination, I guess. Yeah, and sharing.
Thank you very much. There's a question in this chat from Zakaria El Machachi, who says, excellent talk, Syma and John. With metadynamics, how difficult is it to set up the biases to ensure sufficient sampling? And is there a robust way to determine this, or is it done by feeling?
Okay, so I'm going to prefix this by saying openly that, you know, I'm not an expert in metadynamics. We have been using it. In our hands, on a range of different problems, it's actually we found it quite difficult to use. Identifying the appropriate collective variable for some problems can be quite challenging. Now, there are automated procedures that are being developed that might be able to help you with that, but we've never tried them. So we've been working primarily with published collective variables. For example, we've been doing a lot of peptide folding, for example. In relation to this project, it actually worked remarkably smoothly. I'm not sure whether Mohammed Ali is able to step in or Khalid is on the call, but they found appropriate collective variables that gave them the sampling that they needed comparatively easily, at least based on our previous experience. So I guess I would say that the right collective variable can be quite difficult to find. I think it can be quite system dependent and experience based. In terms of ensuring efficient sampling, then ideally one should, of course, once the biases are in place, one should be getting uniform sampling across the entire collective variable. So that is a way of determining whether or not your collective variable is working smoothly, or whether there are slow degrees of freedom that you haven't explicitly coupled in that could be limiting your sampling. Again, in this case, it all seemed to work quite smoothly. But I know for a fact that isn't always the case. I hope that answers the question. In our hands, it's system and context dependent.
Great, thank you very much for that answer. Given the time, I think we have time for one more quick question if anyone sends it in quickly. Otherwise, while people are writing up, I'd like to take this opportunity again to thank you both for giving these two very, very interesting talks and for taking the time to show the work that Biosim has been doing, like the amount of research and work that Biosim has been doing, and going into specifics of the metadynamics work that's been going on. I've genuinely found this fascinating, and thank you both for taking the time to come here and give these presentations.
No, thank you.
Thank you.
And I suppose there's no more questions at the moment, so I believe that we will finish the webinar there.
Okay, thank you, everybody.
Thank you, Julie. Thanks, everyone.